In the biopharmaceutical and medical device industries, endotoxin detection is a critical quality control process. And for over 40 years, the gold standard for this testing has been the Limulus Amebocyte Lysate or LAL test, which is derived from the blood of the Atlantic horseshoe crab. But a confluence of environmental, economic, and scientific pressures is driving the industry away from this long-standing standard. Today, we'll look at the market for endotoxin testing, the challenges associated with the current state of affairs, and the novel IP that is enabling a transition to a more sustainable and secure future. The global endotoxin testing market was valued at 𝗕𝗟𝗢𝗢𝗡𝗧𝗏 billion in 2022. It is growing at approximately 4% annually and is expected to reach 𝗕𝗟𝗢𝗢𝗡 billion by 2024. Key growth drivers include the continued expansion of biopharmaceutical production, stricter safety regulations, and increased biologics and vaccine development. While the market today is relatively small, it's highly specialized with a few large players dominating. By far the largest supplier of endotoxin testing products and services is the Swiss company Lonza Group, which holds approximately 35% share of the global LAL market. Lonza is also the market leader in synthetic recombinant factor C or RFC, which represents the most mature and widely adopted alternative to traditional LAL, in addition to its Purigene RFC assay. Lonza offers a range of other endotoxin detection solutions, including sample preparation products, instrumentation, and services. Other key players in the space include Charles River Laboratories, which supplies EndoSafe LAL cartridges and is also developing and marketing Recombinant Cascade Reagents or RCR as a next-generation replacement for LAL. Then there's Associates of 𝗕𝗡𝗗𝗢𝗏, one of the oldest LAL producers, which is now a subsidiary of the Japanese company Seikagaku. Despite being engaged in conservation efforts, ACC still sources its LAL from wild crab blood. The industry is moving towards synthetics. The most promising alternatives to LAL are based on recombinant technology, specifically RFC and RCR. Recombinant Factor C or RFC was the first synthetic method to gain widespread adoption. It uses genetically engineered protein that mimics the endotoxin-sensitive component of LAL, but without the need for animal blood. This approach eliminates batch variability, reduces manufacturing complexity, and enhances supply chain security. Building on this success, Lonza has developed its own Recombinant Cascade Reagents or RCR, which goes beyond RFC by fully replicating the enzymatic cascade of Limulus Amebocyte Lysate synthetically. This innovation allows for broader application compatibility and paves the way for a full replacement of LAL. Aquaculture-based approaches are also gaining traction. One such company is 𝗕𝗡𝗢𝗢𝗡𝗧𝗏, which is exploring the use of crab aquaculture to produce LAL. This innovative solution could help alleviate pressure on wild crab populations while providing a sustainable source of horseshoe crab blood. Meanwhile, other companies are working on entirely different approaches to endotoxin identification, such as a biosensor platform or a cell-free detection system. These new tools offer rapid and sensitive detection of endotoxin, potentially transforming the efficiency and speed of endotoxin testing. The transition to synthetics is well underway. For many years, the pharma industry has been moving towards incorporating more sustainable and secure sourcing practices. In this context, the recent update of pharmacopeal standards, such as the USP Chapter 86 𝗓𝗏, is a significant development. The new guidelines now explicitly recognize and validate synthetic methods for endotoxin testing, paving the way for their broader adoption. The transition from LAL to synthetics is expected to take 5 to 10 years, driven primarily by pharma ESG goals and supply security requirements. However, some forward-looking companies are already incorporating next-generation synthetic IP into their manufacturing processes. The opportunity for early adopters is clear. Manufacturers that are heavy users of LAL are exposed to a range of environmental, regulatory, and reputational risks. Proactive companies are seeking to mitigate these risks by investing in and validating synthetic testing platforms that offer not only sustainability benefits, but also enhanced sensitivity, specificity, and supply chain resilience. By doing so, they are not only future-proofing their operations, but also differentiating themselves from competitors and positioning themselves at the forefront of this industry transformation.